Greetings from LML, Lingam Microbiological Laboratory. We are a specialized manufacturer of antimicrobial susceptibility testing devices proudly based in India. In this video, we will demonstrate the gradient diffusion method, a simple and reliable technique to determine the minimum inhibitory concentration, that is MIC of antimicrobial agents in a microbiology laboratory. Like the standard disk diffusion method, the materials and procedures remain mostly same. However, in this test, we use the MIC strip instead of a disk, allowing us to measure the quantitative susceptibility of microorganisms, that is, the lowest concentration of an antimicrobial agent that inhibits visible growth. The MIC strip is impregnated with the continuous gradient of an antimicrobial agent along a predefined scale. Once placed on the agar surface, the drug begins to diffuse exponentially, forming a concentration gradient that interacts with the bacterial growth. For more information on materials required, we recommend watching our disk diffusion method video. Click the eye icon on the top right corner or find the link in the description below. Let's begin the procedure. First, sterilize the inoculation loop by holding it in a flame until it becomes red hot and allow it to cool for a few seconds. Then, pick pure colonies from a culture plate, preferably from a nutrient agar or blood agar, and suspend them in sterile saline or nutrient broth to prepare a bacterial inoculum. Adjust the suspension to match 0.5 McFarlane turbidity by adding either more colonies or diluent as needed. Take a pre-dried sterile Muller-Tintin agar plate into a sterile workspace. Label the bottom of the plate with relevant information like date, sample number, or organism name. Using a sterile cotton swab, dip it into the bacterial suspension and then remove the excess fluid by pressing the swab against the inner wall of the test tube. Inoculate the agar surface by swabbing evenly in three directions, rotating the plate about 60 degrees between each streak to ensure the uniform lawn. Finally, run the swab around the plate's edge and discard it into a disinfectant jar. Place the agar plate upright on an MIC strip positioning template. Choose the appropriate template based on the number of strips you intend to apply. Using sterile forceps, carefully hold the MIC strip by the top edge bearing the logo. Ensure that the printed side faces upward. This side includes the concentration scale and identification markings. The blank, porous underside is the active antimicrobial impregnated surface, which must be in direct contact with the agar medium for proper diffusion and accurate MIC determination. Begin by gently placing the lower end of the strip, which contains the lowest concentration of the antimicrobial, onto the agar surface. Then slowly roll the rest of the strip down, allowing it to make full contact with the agar along its entire length. Ensure that the strip lies flat and firmly adhered to the medium without any air gaps or lifting. Do not move the strip once it touches the medium, even if placed at wrong position. The antimicrobial immediately starts diffusing at the point of contact, and repositioning it will distort the results. Incubate the plate inverted at 35 degrees Celsius for 16 to 18 hours. After incubation, examine the plate for bacterial growth and an elliptical zone of inhibition around the MIC strip. The MIC is read at the point where the inhibition ellipse intersects the MIC scale printed on the strip, expressed in microgram per milliliter. If the ellipse intersects between the two values or asymmetric, showing different values on each side, read the higher concentration as the MIC. However, if the discrepancy exceeds one doubling dilution factor, repeat the test for accuracy. Interpret the MIC result using recognized standards such as CLSI, EUCAST, or local guidelines, and classify the organism as susceptible, intermediate, or resistant. At LML, our MIC test strips are marketed under the brand name LingMIC, featuring standardized 3-letter antimicrobial codes compatible with WhoNet software, ensuring smooth data integration and reporting. All our products are manufactured under strict quality control protocols, offering high precision, reproducibility, and longer shelf life. Our devices are trusted by clinical laboratories, academic institutions, and research centers worldwide. Thank you for watching this demonstration of the MIC gradient diffusion method. Don't forget to like, share, and subscribe to our channel for more microbiology tutorials and product insights. For bulk orders or technical inquiries, visit our website or reach out to us directly. We look forward to serving your laboratory needs with precision, reliability, and trust.